think we'll get this started because we just like to stay on time and make sure you have lots of opportunity for questions later. So my name is Peter Unrau, and together with Nancy Ford, who is over here, we are the organizers of the President's Dream Colloquium series for this fall, together with innumerable other people that have been making everything go as smoothly as possible. So I'd just like to say a few organizational things, and then I'll hand the mic over to Dave, who's going to introduce Bill Schopf, who's our speaker for today. So the basic key points are that there's a reception right after. Please go to it and mingle. Bill will be there. And at 5:30, if you've been invited to dinner, we'll be leaving from the reception to go to dinner. If you're interested in the philosopher's cafe,
I'm over 60.
Mike Picard, where is Mike Picard, will also be going to the pub at 5:30. So if you're interested in that, please join Mike. Okay, so without further ado, I'll hand the mic to David.
Peter, this is not good, Peter. Okay, thanks very much, and thank you all for coming today. It's a real pleasure to welcome Bill Schopf and his wife Jane, who's here with him today, to SFU. This is not his first time here. I guess 2 years after he graduated with his PhD from Harvard, he came here and gave a talk in 1970. So Bill is one of the preeminent figures in paleobiology. His academic grounding is very thorough in both areas. He did his undergraduate in geology and 2 degrees in biology from Harvard. His accomplishments are many, have been well recognized in terms of awards. The things I would like to point out today for you are, this is the Old Testament or this is the New Testament? This is the Old Testament on which he edited with and made heavy contributions to on the Earth's earliest biosphere. And this thing, which I had to pay an absolutely ridiculous amount of money for from Cambridge when it was reprinted, is the New Testament. Hans Hofmann is, I think, a co-editor on... Was he a co-editor on any of these?
He was a member of both groups.
Right, okay. Who is from McGill, from University of Montreal, and then later an associate member of McGill, was also one of the contributors. On the popular side is this tome, The Cradle of Life, which I've certainly recommended to many people and which my wife has read as well. So without further ado, Bill, there's one other thing I should mention. Not only did he give a talk here very early on, but he was also the photographer at Lalit Srivastawa's wedding, for those of you who know Lalit. Okay, welcome.
Thank you. Thank you. It is a pleasure, of course, to be here once again after some 40 years. And a real pleasure for me to see how Simon Fraser University has grown from 5,000 to 25,000, 5-fold increase. This place is remarkably vibrant and remarkably successful, and I think you're all really doggone lucky to be here. So, and I'm fortunate to be here. Okay, moving right along, he says, if he can figure out what he's supposed to do. Okay, how do I go back one slide? It goes back this way. No, it doesn't go back that way. Which one? Okay, I'll use that. I'm going to talk a little bit today about the earliest history of life, solution to Darwin's dilemma. Now, as most of you folks must realize, that a great amount of modern natural science comes from the work of Charles Robert Darwin. And of course, he is perhaps best known for his 1859 book on the origin of species. And in that book, he devotes at 12 different places a discussion of the missing early record of life. He says, if the theory of evolution be true, it's indisputable that before the lowest Cambrian stratum was deposited, the world swarmed with living creatures. Yet why we do not find these rich fossiliferous deposits belonging to these assumed earliest periods, I can give no satisfactory answer. The case at present must remain inexplicable, and may be truly urged as a valid argument against the views here entertained. He was really puzzled by the lack of a pre-Cambrian fossil record. And in Darwin's time, this was known. In fact, it would really had been known since the early 1800s, the age of invertebrates and of fish and dinosaurs and mammals and so forth. But the oldest fossils known were from the lowest Cambrian strata, now dated around 550 million years ago, and organisms such as this, trilobites. In that particular trilobite, the genus Olanellus. This was Darwin's problem. If evolution were to make any sense, there had to be a long fossil record preceding these very advanced, multicellular, complicated organisms with eyeballs and mouths and snifters and all that good stuff. But it was not known. So he presents the problem. We then go on up to the present, and this is what we now know, that the Precambrian was the age of microscopic life. No one had predicted that. No one knew that. It was something that has been discovered in the last 50 years. Well, Darwin states the problem in 1859, and you would imagine, therefore, that Darwin, who was, remember, at the seat of Western intellectual activity, London, the British Empire owned the world in 1859, and you would think that everybody listened to him, which they did. And people would go out there and they'd find these early fossils. And they tried, and they tried, and they tried, and there were innumerable mistakes made, none of which held up. And in fact, there was a hiatus of about 100 years up until the mid-1950s before any meaningful progress was made. Now you ought to ask yourself, I wonder why that was. This was a good problem. It sat there for 1 century, more than a century, but no progress was made. Why was there no progress? The answer is that everyone, including Darwin, and all of his contemporaries and all the generations, the 4 or 5 generations that followed, made the same error. They assumed that Precambrian life would be like that in all of the younger strata that had been well studied, and increasingly well studied, and studied up to today. It turns out that this was a wrong assumption. The wrong question was asked. It wasn't until the 1950s, in the mid-1950s, that the tide began to change. And it began to change first in the Soviet Union. And I personally do not think that the Soviet workers get the recognition that they deserve. But the key active person there was a man named Boris Vasilyevich Timofeyev, at the Institute of Geology, Precambrian Geology and Geochronology in Leningrad, now St. Petersburg. And what Timofeyev, this gentleman here, had done was to discover that if he were to take shales, siltstones, shaly rocks, very fine-grained rocks, dissolve them in mineral acid, hydrofluoric acid, concentrate the organic matter within them, something that palynologists do routinely today, but this was new stuff in the early 1950s, which he developed to be used in the Soviet petroleum industry, that in these shaly rocks there would be microscopic single-celled organisms such as those shown here. Now, Timofeyev made some errors. He was not a biologist, didn't have any training in biology, He interpreted these to be the spores of or pollen grains of land plants, mostly the spores of land plants. But land plants don't arise until 420 million years ago, and these rocks were back to 1,000 million years ago. So that didn't make any sense. Nevertheless, it turns out they're perfectly good fossils, and we now know that they were phytoplankton that floated in the Precambrian seas. Well, that was the first big breakthrough. Wasn't recognized for various political reasons, primarily in the West for quite some time. And then came along Stanley Tyler at the University of Wisconsin, an economic geologist, and Elso Barghorn, a paleobotanist and botanist at Harvard University. And here is Tyler on the left. Tyler discovered microscopic fossils in a deposit that many of the students here know about, the Gunflint Chert on the northern shore of Lake Superior. He discovered those in 1953, and through a series of serendipitous interactions with various people, by 1954 at the Geological Society of America meeting in Boston that year, he was introduced to Elso Barghorn, the new paleobotanist at Harvard, and the two of them wrote a short paper in 1954, and then finally, it was a 2-paragraph paper with a certain number of mistakes in it, and finally in 1965, they published these, and this is what you will see in normal textbooks. Sets of little single cells and these long filamentous objects. And that's the sort of thing that I understand that last week students had an opportunity to look at some thin petrographic slices of rock and with a microscope, and they could see. This is what you saw. What you didn't see, excuse me, and often don't see even in textbooks, is this particular organism. I'll bet you didn't see it because I've only seen about 150 of them and they're relatively rare. It's an unusual sort of thing. It's called Eosphaera. It's got an inner sphere and then these little spheres on the periphery and then an outer sphere. It looks rather like a volleyball with a set of golf balls enshrouded by a basketball. Kind of a bizarre thing, does not live today. It went extinct, not too surprisingly, because let us remember the trilobites are extinct, the dinosaurs are extinct. Extinct is extinct. That's what you'd expect, you know, a bunch of stuff that has been there, done its thing, and is no longer around. Another one, particularly interesting, that folks talked about at lunch today is Cacabeckia umbellata. Has a sort of a little ball down here and an umbrella on top. Is kind of like a chocolate-covered peanut with a Japanese umbrella. That's another one that is a perfectly good fossil that is extinct, and if I had time, I could show you its life cycle, even though I don't know what it's related to today. Okay, well, that's the Gunflint assemblage. That's a brief history of this field. And what is interesting about it is it doesn't matter what you plot on the ordinate here. That's the history of the field. Number of species named, publications, discoveries, workers, time spent, money, whatever. It all goes up, starts 1965 and comes up to the present. And now there are several hundred workers worldwide that have found many hundreds of microscopic assemblages throughout the world. And we're having an awful lot of fun trying to fight through the history of life and figure out what was going on in the earliest 7/8 of Earth history. Okay, that's the history of the field. Now, where does that information come from? It comes from 4 different lines of evidence, things called stromatolites, microscopic fossils, molecular biomarkers, and carbon isotopes. Because of time constraints, folks, I'm only going to talk about stromatolites and microfossils here. If anybody wants to hear me harangue them about the other stuff, I'll be pleased to harangue. I usually harangue in 50-minute segments. I'm a professor. Now, with regard to stromatolites, what are they? Look, this is, you guys know that most everything that, all these scientific terms, they're derived from ancient Greek or ancient Latin, okay? And here we got stromatose, which means bed covering or layer, and then the linking vowel that goes into the other root, lithos, which is used in geology, lithology, the study of rocks. So, stromatolite is a layered rock, but it's a curiously layered rock because it is layered by the activities of microscopic organisms. One of the best known modern examples comes from Shark Bay and Hamlin Pool in particular on the western coast of Western Australia. And here are a bevy of stromatolites, these things like that. They're made out of calcium carbonate. You can walk. Well, you can't now. It's all off. It's protected. You're not allowed to. But in the early days, you could walk across these things and get up and have a good look at them. If you look at them up close, you'll see that they have a kind of a bluish, greenish surface layer to them. And if you looked at that with a microscope, you'd see it was made of organisms of this type, cyanobacteria. Now, cyano means blue-green. Bacteria means bacteria. And they are green because they have chlorophyll A within them. These are the organisms that invented oxygen-producing photosynthesis. Their biochemistry to do photosynthesis is exactly the same as one of these nice trees on your campus, or a blade of grass, or a corn stalk, or anything that's green. They all do the same stuff, and these guys are the guys that invented it hundreds of thousands, or thousands of millions of years ago. Okay, well, here at when the high tide was in from the Indian Ocean on the western coast of Australia is a bunch of the Shark Bay stromatolites, and here is the equivalent, 2.3 billion years in age from South Africa, the Botsap River section in particular. And these things go right back through time, back to 3.5 billion years ago. They're found in always within the photic zone because they're photosynthesized, formed by photosynthetic dominated communities of microorganisms in places where carbonate rocks are deposited. So here is a vertically sliced section of one of the Shark Bay stromatolites. Here is the equivalent from the Belt supergroup, around 1.3 billion years in age from Montana. And here's the equivalent at 3.2 billion years in age from South Africa. So those are stromatolites, major evidence of life, and probably the world's expert on really old stromatolites, certainly the world's expert, was Hans Hofmann. His name's been mentioned here before at the University of Montreal and then later at McGill. And Hans did enormous, wonderful work throughout Canada, discovering and showing the history of these stromatolites through time. Well, you ask, what good are stromatolites? Now, come on. I mean, you always sit there, okay, I've never heard of a stromatolite. Who cares about a stromatolite? What good are stromatolites? Who even likes stromatolites? They make good doorstops. But I want to take you on a little trip to Beijing, China, but of 30 years ago, 1981. This is my wife, Jane, right here. And as you see, she looks just the same. She's right over here. And our son Jamie, and we're in front of the Friendship Hotel there in Beijing. And if you walk into that hotel and you go to the registration desk, you'll see this. Well, you won't see this, but you'll see sort of this. This is 1981, but I really want you to see this registration desk. That is stromatolites. Stromatolites all over the place. You look on the floor, there are stromatolites. You can see that's my wife's toe for scale. And you can see the layers in them. They're just beautiful. They've been used in Chinese architecture since at least 4,000 years to my knowledge. They're often used as facing stones, and they're really pretty, and they're really nice if you happen to be interested in them, like me. Well, one day we were at Tiananmen Square, and this is the Great Hall of the People, and I spotted this from, I don't know, a half a mile away. That square is huge. And I said, I bet you those are made out of stromatolites. So I wandered up there and got my camera out so I could take some pictures. And here, this is just all stromatolites. This is the Northeast Red Formation. It's 780 million years in age, comes from near Dalian, which is up near what used to be called Manchuria. And this is the Wumasan Formation. It's 1.5 billion years in age. It's a place that I was fortunate enough to find the first ancient fossils of this sort in China. And then the Chinese police came and they said, you're not supposed to be here. and the other two guys who were with me from Nanjing Institute of Geology and Paleontology, they had disappeared. And I said, I don't speak Chinese and so forth. And so I was escorted away, but I got the picture, And they did not put me in jail. They were very nice to me, and I got away. Then a few days later, I was riding my bicycle. This is 1981, and in China, everybody rode a bicycle in those days. And so, of course, I was riding my bicycle to the institute where I was working, and I happened to see a people's park off to the side. So I went over to this people's park to have a look at it. I don't know why, it just sort of caught my eye, and turns out this slide is made out of stromatolites. It is. And that's the Wumasan again. It's 1.5 billion years in age, has good microscopic fossils in it. I did not take a sample. I simply took a photograph and then went home. Okay, those are stromatolites. Microscopic fossils. Microscopic fossils, the most abundant fossils in the Precambrian are cyanobacteria, the ones that build those stromatolites. Now, here is the equivalent of a stromatolite. These are called microbial mats, and there's a lesson about this. Stromatolites were not understood until 1961. They had been described in 1837. But there had been a long discussion as to whether they were biological or not biological and so forth in the geological literature, until the ones at Shark Bay were first discovered by Philip Playfair in 1960, and then the first paper came out by Brian Logan, a graduate student at the University at Perth, in 1961. Well, that's a shame. I mean, that was great. They were discovered by geologists, but you see there's a real error here, because microbiologists had been working on microbial mats since the turn of the century, since the early 1900s. They didn't call them stromatolites, they called them microbial mats. Here you see these mound-shaped microbial mats, and if you look at them in cross-section, you can see, yes, they have all these laminae to them and everything like that, just like regular stromatolites, and that's in fact what they are. They're the unfossilized equivalent. The lesson here is that the geologists did not read the microbiological literature. The microbiologists, being in the life sciences, did not read the geological literature. And because of this, science sat there for 50 years arguing about these things when the answer was right before their eyes. Now, you students, do better than we professors. No kidding. I mean, learn from us. We'll do our best never to mislead you, but you can do better than we can. You cannot make these errors. This is an interdisciplinary course that you guys are taking. Be interdisciplinary, for gosh sakes. Nature is not compartmentalized. It's not physical sciences and life sciences. The whole mess is there. So learn about the whole mess. Okay. I get worked up here. Now, Here is then a fossil stromatolite. This is indeed a fossil stromatolite from the belt supergroup, 1.3 billion. Here is the modern equivalent living today. And you see how similar they are. Now, if you look at the surface of that modern stromatolite, you would see that it was inhabited by, populated by cyanobacteria. It turns out there are four main groups. Now, we biologists have these fancy names. They are polysyllabic. They have many, many letters to them. We love to ask them on exams because nobody can remember the Croacaceae, Antophysalidaceae, Nostocaceae, and the oscillatoriaceae. Okay. Hey, but some of you guys, are geology types, and some of you guys are molecular biology types, and you don't want to hear about the Croacaceae. So, look, why do we name things? We name things to communicate. So I'm going to give you a different classification. We're not going to call these guys here the Croacaceae. We're going to call them the balls because they have a ball-shaped skulls. These guys, we're going to call them the jelly beans. That's the Antophysalidaceae. These guys down here, we're going to call them beads because they're like beads on a string. That's the Nostocaceae. And these guys are strings because it's strings of cells. That's the oscillatoriaceae. Those are the four main families that build modern stromatolites. Those are the four main families that you find in the fossil record. Okay, so I'll give you a few examples just so that you know that I actually can convince you they exist. Here, these are balls, Croacaceans, 1, 2, 3, 4 cells with a polysaccharide sheath around it. Here's the equivalent, 1.5 billion years in age from Bashkiria, 1, 2, 3, 4 cells, and here's the sheath around it. And there are quite a bunch of examples of that. Here's another example. These are cells of those Croacaceans, those balls, but this is a 4-cell colony, and this is one of the problems we have because to show this, it's a 4-celled colony, you have to take a picture at the top of the colony, and then you take one a little bit down farther where these medial cell walls are in shape. You can see the 2 cells beneath it, and here you get the bottom cells, and here's the base of it. Well, that's not really very good because you got to publish 4 pictures to show that, and a lot of editors don't like that. So about 6 years ago, we started using a new technique called confocal laser scanning microscopy. I stole this from biology. Biologists put dyes that will fluoresce when you put a laser on them, but it turns out the organic matter in the cell walls of these ancient fossils are made of polycyclic aromatic hydrocarbons, and in fact, just like I figured, if you stick the right wavelength of laser on them, you in fact get a nice fluorescence of them inside the rock, okay? And as a matter of fact, then you can make videos and you can spin them around and look at them, and we do a fair amount of this nowadays, and it has been very helpful because you can see the real three-dimensionality of these fossils inside rocks, and this is something we're going to use when we get samples back from Mars, if there's anything in there worth looking at. Okay, jelly beans, the Antophysalidaceans, the modern on the left, and here that's Hans Hofmann's work, as a matter of fact, 2 billion years in age from the Belcher Islands in Hudson Bay. And here, Nostocaceans, the modern bead cells, and here you see the fossil equivalent from Australia, 800 million years in age. And there are lots of examples of this sort. I just wanted to show you those 4 in particular. And then here's some strings, the modern ones above, and here's the fossil, 950, nearly 1 billion years in age. You can see the disc-shaped cells, just as you can see in the modern, the rounded terminal cells, just as in the modern. And here, this is a polysaccharide tube or sheath that surrounds that cellular trichome. And here you can see a bit of it, and here's the rest on the other side of it, and these are sheaths back behind there. There's one other example of strings that I have to show you, and I put this in for you students. I don't know a lot about Simon Fraser, University. I don't know a lot about Simon Fraser either, but I don't know except he was a great explorer. But I do know one thing about universities. And one thing about universities is that throughout the world, on the periphery, there will be health food stores. And those health food stores, they are there for the students. You students, you go to these health food stores, and they charge you these exorbitant prices for this stuff, ginkgo and spirulina. And you never see a faculty member in there. There is a reason for that. We gave up. But you guys, you haven't given up. You are going to keep healthy and all that, and good for you. Well, spirulina is one of these real famous ones that you can get at the health food store right around the corner, folks. You can see here, you get books on it. Yeah, and if you eat enough of it, you can run down the street in your swimming suit. You can get it in pills. You can get it in powder. You can brush your teeth with them. It makes them green. You can get it in candy bars. You can. I don't make this up. You see at the back, the top up there? Rebirth of ancient sustenance. Yeah, that's because they found out that I'd found the same thing, 1 billion years in age. So they don't give me any royalties on this, but there it is. And then if you look at the bottom, see this down here, look real carefully and you can see 22 amino acids, 2 extra. There it is. This is good stuff. Okay, well, and here's the modern and the fossil, okay, 850 million years in age. Okay, well, that's cyanobacteria. The oldest evidence of life comes from northwestern Western Australia and from southern Africa, particularly near the kingdom of Swaziland, in rocks that are 3 to 3.5 billion years in age. There are lots of stromatolites in both localities. Hans Hofmann, to my knowledge, described from Canada, from South Africa, from Australia, and in fact, these are Hofmann specimens right here, 3.5 billion, or nearly that, years in age, from 42 different localities, 42 different geologic units. So they're very well known and widespread. There are some 22 microfossiliferous units between 3 billion and 3.5 billion. One of the best known ones and most studied ones is shown here from northwestern Australia at 3.465 billion years. You can see these cellular filaments. These are Raman spectra that show that the organic material of which these are made is not graphite. It's the organic material that all permineralized fossils of this sort are composed. We also show that they're carbonaceous by using confocal laser scanning microscopy, and you can see that they fluoresce. They're made out of polycyclic aromatic hydrocarbons, not graphite. They are, in fact, they're also cellular. They're cylindrical, as you can see in cross-section. This is a three-dimensional Raman image, something that we pioneered in my lab. And then you can strip away from these three-dimensional images layer upon layer. So here, down 3/4 of a micron down into the specimen, you can see these cells. Now we're down to 1.5 microns, 2.25, 3, down to. You go right down through that and see that indeed they're made out of carbonaceous material that defines these box-like cells, just like all of the other younger Precambrian fossils that we've studied. Okay, so that's the history of life. Now, I have cleverly left myself some time, because now I want to regale you with some stuff that's not yet published. and something I'm particularly pleased about. And a brand new ecosystem has been discovered, and I think this is the first time that I've actually given a public talk in which I've shown these things. And it'll be new to you, and you'll have questions, and then you will be convinced. What are these things? Well, they are non-photosynthetic, sulfur-metabolizing, anaerobic microbes. An ecosystem called a sulfuretum, and that name will be new to you, many of you, and I'll show you just what that is all about. But I wouldn't waste your time with just one, because I got two. One at 2.3 billion years in age, and another one at 1.8 billion years in age. And what's particularly interesting about that is that this is the time of what's called the Great Oxidation Event. And what has happened is that well before that time, there are cyanobacteria. Cyanobacteria are photosynthesizing and pumping oxygen into the environment. A lot of that oxygen is being sucked up, reacted with ferrous iron to make banded iron formations and iron-rich rock. After a while, the iron reservoir is used up, so the oxygen starts building up. And now, largely through the work of Dick Holland, but a whole lot of other people, it's now very well established that this great oxidation event happens between about 2.4 and 2.2 billion years ago, and that was a major increase in the major change in the Earth's environment, because with that oxygen, you see obligately aerobic organisms such as ourselves that cannot exist. You cannot exist for more than 6 or 8 minutes without oxygen. It all comes from that great oxidation event, okay? So that's where we are in Earth history. And I claim that there are two of these guys that we've found, one of which is in a paper now submitted to Nature, and the other one, the manuscript is finished, but I don't want to put it in until we get the first one published. So, what is a sulfuretum? This is the work of Victor Gallardo and his wife Claro Espinosa at the University of Concepcion in Chile. This is off the western coast of Chile. Here that yellow thing is labeled sulfuretum. What is a sulfuretum? Well, here's how it works today. Seawater sulfate comes into this system. There are sulfate-reducing bacteria that live by reducing that sulfate to hydrogen sulfide. Okay, they're very well known, and we can see their presence by sulfur isotopes in pyrite, things like that. Okay, well, if there's reduced iron around, ferrous iron, that iron reacts with the hydrogen sulfide to make pyrite. And it has a characteristic isotopic signature, so we can identify that. If, however, there is an excess of this hydrogen sulfide, then another set of organisms that are anaerobic, living in the absence of oxygen, can oxidize using nitrate from seawater up to elemental sulfur. And there is yet another set of sulfur metabolizing things that'll take that elemental sulfur and anaerobically oxidize it back to sulfate. So it's a very simple little system of going from sulfate to hydrogen sulfide to sulfur, and around it goes. And these three different sets of organisms, and these organisms get energy because, you see, there are 8 valent states to sulfur, and so it's a redox set of reactions. And so by shunting around those electrons, they're getting energy for themselves. All right, so I claim I've got 2 of those. How do I know that? Well, one of them is 2.3 billion years in age. It comes from here in northwestern Australia. Now, first I got to show you that they are not stromatolites, because that's what everybody has always found heretofore before this discovery. Well, what does a stromatolite look like? You can see they're layered, and here is a thin section, petrographic thin section of a stromatolite, and here is some more at increasing magnification. And you see these little filaments here. In stromatolites, the microorganisms' filaments are aligned parallel to the layers, and that's what causes the layers. Big deal. Well, we all know that. This is the fabric of the sulfuridum. It's just like the modern sulfuridum indeed as well. Notice you got this mottled fabric. Now you look at this and look at the filaments. They're completely random. They're a big cobweb of mix, which puzzled me for quite some time because I'd never seen this in the geologic record and nobody else had either, and I didn't know what to make of it for a while. Okay, so the fabric is really different. And you see, what the microorganisms in megastromatolite are spreading across surfaces because they're photosynthetic. These guys are not photosynthetic, so they're all mixed up. Okay? That's first line of evidence. Now you go to the geology. See, this is this interdisciplinary science. It's not microbiology, it's not paleontology, it's not geology, it's geology, it's isotopes, it's organic geochemistry. All this stuff has to come together to make sense out of this. So you go to the field and you look at it and you say, oh, where does this fit in stratigraphically? Well, you look at that and there are a lot of things like ripples and desiccation cracks that tell you, but all of them are absent. This is deep water. And then you do what's called sequence stratigraphy, and you know exactly where this fits in, and it's real deep water stuff, and it, as a matter of fact, looks like it's beneath photic zone. And then you go to over here, and you look at where the fossils are. They're in a thin-bedded marl, which tells you it's far from sedimentation. You're offshore someplace or other. Okay, and now you look at this cobweb-like fabric that you've never seen before. It's not laminated and layered like a stromatolite. And now you look at the filaments and you see, gosh, these things are hundreds of microns, some of them thousands of microns in length. Now that makes no sense. You never see that in a stromatolitic assemblage. Why? Because stromatolites are up near wave action. They're places where the filaments are getting broken up because the waves are going up and down and up and down. This stuff is at the bottom of the ocean. It's quiet. There is no wave action. They're in the muds at the bottom of the ocean. It's dark, it's cold, and you couldn't photosynthesize down there because there ain't no light. All right, so now you say, well, that's interesting. All that stuff gives you those hints. What do the organisms look like? Well, there are four major types, both in the modern sulfuridum and in the fossil one. Here's the modern one. These have long cells around 10 microns or so. They're 10 microns in diameter. Here's the fossil equivalent. That's the modern in the blue, the fossil beneath it. That's one type. A second type with this intermediate sort of diameter bead-like cells that you can see. Well, here's the fossil equivalent. Same size, same shape, same organization, nice and long, just like the modern ones. These are more tiny bead-shaped ones, and here's the fossil equivalent. And then you look at the real long, very thin, narrow, less than a micron in diameter, and here's the fossil one. So you've got the same environment, the same fabric, the same setting, and the same organisms between the modern ones and these guys at 2.3 billion. Well, now I said that there was a second one of these guys. So let's see what it looks like. Well, it comes from the next geologic sequence to the northeast there in Australia. And it turns out it's called the Duck Creek. And just like the geology is absolutely the same, there's no evidence of any shallow water stuff. You know you're deep water because you're off a shelf, and the rocks are, again, this carbonate marl. Here is the thin section and the fabric of that 2.3 billion year old rock. Here's the Same thing at 1.8. And I will tell you, folks, if you take those two hand specimens and you hold them side by side, they look exactly the same. And here is the same fabric that has never been seen before in the geologic record, at least has never been reported anywhere. But you notice also, you see the filaments all messed up, all cobwebby, the same exactly, the same old thing. And it turns out that these white areas in here turn out to be domains where there was anhydrite that was being precipitated due to the rise of sulfate in these particular environments. Well, here's the fossils you've already seen. Modern, 2.3 billion. Here, 1.8 billion. Exactly the same organisms. Here, the intermediate diameter microbes, the modern one, 2.3. Here's at 1.8. Exactly the same organisms. Here, the narrow microbes with bead-shaped cells, modern 2.3. Here at 1.8. And here's the fourth type, modern and 2.3, these very long, narrow microbes, and here's the equivalent at 1.8. So it's the same fabric, same geology, same preservation, same organisms, same depth, same deal. Now that's telling us something. Not only that, there are other evidences that tell you that these biotas are anaerobic. In the first place, the modern equivalents live in anaerobic mud, where there is no oxygen beneath the surface, the sediment-water interface. Secondly, this fabric and these very long filaments tell you that it's got to be a quiescent environment. You're in the subsurface. You're in an anoxic zone where the muds are not being churned up. Remember, there are no metazoans around at this time to do bioturbation. These are microbes that are just sitting there minding their own business in these muds. Thirdly, there are pyrite-replaced microbial filaments, and that tells you can't get that unless you've got an anoxic environment. And you say, how do you know they're pyrite-replaced? Well, because we do Raman spectroscopy. Here are the filaments. Here we show that they're made out of organic matter, and here the red stuff, that's the pyrite of which they are composed, and you can see there's a whole lot of them. Now, the waters directly overlying these sulfurida were dysoxic, which means that they were really, really low in oxygen, if they had any oxygen at all. How do you know that? Well, first of all, this was a time in Earth history where the upper portions of the water column in the world's oceans had some oxygen in them. But as you go down, it's what's called a stratified ocean, or the Canfield Ocean, and there's lots of pyritic-rich black shales being deposited all over the place. Tells you that the lower waters were anoxic. But not only that, the biology tells you that. There's a particular organism known as Eoastrion. Its modern equivalent is called Metallogenium, and it occurs, for example, in Lake Washington. Here are the modern organisms that live today. They oxidize, they precipitate manganese oxides and iron oxides and titanium oxides. Well, here, this is an optical picture. Here is a Raman picture. It shows it's made out of organic matter, of this kerogen. That's at 2.3 billion. Here's the same thing at 1.8 billion. And we can do Raman spectroscopy on those that have been mineral encrusted, just like the modern ones. And here, oh gosh, organic matter shows it's biological. Here's Raman. This is pyrite after iron oxide, and here's your titanium oxide getting precipitated. So that doesn't happen unless you are in a place where there's these are plankton that are floating down into the deposit. It does not happen unless you're in a place where there's hardly any oxygen at all. So that just nails the thing. It tells you a whole lot about the ecology. See, this is not just rocks and this is not just shells. This is not just paleontology. This is microbiology. This is biochemistry. This is the whole stuff has to come together to make any sense out of it. But this makes two big questions that ought to be niggling at your minds. One, how does this fit Earth history in this great oxidation event? And two, what about this amazing lack of evolution? Okay, well, remember what the sulfuridum does. It needs these three reactants, sulfate, hydrogen sulfide, and nitrogen. Well, before 2.4 billion years ago, there wasn't much sulfate in the world's ocean. It was present, but not in particular abundance because there was hardly any oxygen in the Earth's atmosphere. The same thing with nitrate. There was hardly any nitrate. Something like 50% of the world's nitrate is formed today by electric discharge because you got nitrogen and you got oxygen and you get lightning. Okay, but there's no oxygen around. You can't make nitrate that way. That's why there's a long story I won't go into. That's why microbes have nitrogen fixation, whereas no eukaryotes do. It all makes good biochemical sense. And there wasn't a whole lot of hydrogen sulfide. Why? Well, because there weren't a whole lot of sulfate-reducing bacteria. They were present, but they weren't very ubiquitous because there wasn't much sulfate. And in any case, the hydrogen sulfide is going to react with ferrous iron because there's no oxygen around, so you get pyrite precipitated. Now you get the great oxidation event. What happens? Major increase in sulfate because there's a major increase of oxygen. Major increase of nitrate because there's a major increase of oxygen. Major increase of hydrogen sulfide. Why? Because there's a major increase in sulfate. Since there's a major increase in sulfate, there's a major increase of sulfate-reducing bacteria that are pumping this H2S into the environment. And remember also this ferrous iron that was there before is now reacting with oxygen to make hematite. So it's taken, the ferrous iron's taken out, so you get less pyrite. So the whole thing sticks together, and it turns out that increased oxygen at the great oxidation event increases sulfate, it increases nitrate, it increases H2S, and those are the three reactants you need to power these non-photosynthetic bacterial sulfuridas, and that's exactly what we have found at that time. Okay, so that all fits together, and now you come to this lack of evolution. And I personally think this is the most important aspect of this, and maybe the most difficult for some of you to swallow, because to a number of you it'll be an absolutely new idea. But I make the claim, as I've just made, that I've got the same biota at 2.3 and 1.8. Notice, that's 500 million years. That is the length of the Phanerozoic. I say they don't change over that length of time, but I make even a bolder statement. I say they don't change over more than 2 billion years. Now, how could that possibly be the case? We all know that evolution is evolution. It's evolving. How could that be? Well, turns out that facts are facts, and that's the way it goes. And, you can't argue with Mother Nature. And these guys lived in a subsediment, ocean bottom environment that has been constant over Earth history, an absolutely new idea to, I bet, everybody in this room, and I wouldn't have thought about it until I started thinking about it because I had to come up with some way to figure out what was going on. The fact is that environment has always been ocean mud. It's 2 degrees Celsius. It is cold, there is no light there, there is no wave action, there is no oxygen. That is a stable environment. Now think about evolution. When you think about evolution, what you're thinking about is eukaryotic evolution. You're thinking about the evolution of metazoans and plants and animals and stuff like that. And you think of speciation. You think of speciation, adaptation, specialization to an environment, and extinction. That's what we think evolution's all about. Well, that's not the way it is, you see, because that is true for eukaryotic, sexually reproducing organisms if the environment is changing. And I mean physical environment, and I mean biological environment. You get new organisms, that makes a new biological environment as well as a physical environment. But now think of the opposite. Think of a flat world. Think of a world in which there is no change over time. But you got a bunch of organisms that are adapted to that flat world. But that flat world does not change. There are no hills, there's no valleys, there's no change in the chemistry of the environment. The thing is just sitting there all by itself. Folks, in an unchanged environment, there is no evolution. And that is what this says, and that's why this little study matters, because this is the null hypothesis that is required by evolution. If Darwin's evolution is right, it must also be true that in the lack of evolution, lack of environmental change, lack of biological change, lack of change in the chemistry and the environment, there cannot be evolution, and that's exactly what we're seeing. And I think that's really cool. I think that's really neat. I'm really pleased about that. My goodness gracious. Now we've had 150 years since Darwin stated the problem. We've had 50 years since Timofeyev, since Stanley Tyler, since Barghorn. What Darwin said was that the absence of Precambrian fossils is inexplicable and may be truly urged as a valid argument against the views here entertained. Well, I came all the way up the coast from way down there in Southern California to tell you that what's inexplicable to Darwin is no longer so to us. Thank you very much.
Holy cats. Bill has won a very large number of awards, and among them is a teaching award, and we can now see why. Okay, questions from the audience. Now, how are we going to do this?
I think they're all shell-shocked. Okay. We'll do it. Yes, there's a microphone. I think it's going to be passed to somebody. Yes, sir.
I just want to know, a guy like you who thinks not in terms of hundreds of years or thousands or even millions, but in billions. I mean, what do you think of this current thing with climate change?
Okay, I think, as I understand it, the question is, what do I think of the current climate thing is? The current climate change, global warming, as I would prefer to call it, but I don't care, it's climate change. I think I'll try to be brief, which is difficult for me. But first of all, it's a fact. It is, in my opinion, not debatable. Second of all, it teaches us the fact that it has been debated and argued by people. It suggests to me an ignorance, a lack of understanding on the part of those people who have not looked at the scientific data. But more than that, a motivation that I think we ought to all be aware of, that people will honestly say, they will believe things and say things that have other motivations. And in this case, the fact is it costs money to clean up CO2. And folks don't want to spend the money. I don't blame them that they don't want to spend the money. I understand that. But that to then say that CO2 is not going up, and let's find an excuse here. I think the data are irrefutable, and I won't go, sea level rise and the biodiversity changes and all this kind of stuff. It seems to me overwhelming. I mean, absolutely overwhelming. Thirdly, I've been asked this by various people. Have we reached a tipping point? I think we have. I think that we are in irretrievable mess at the moment. I think that at the moment we are being confronted with a problem that we're going to have to try to fix. And it's very difficult. As you see, we human beings are not very good at coming together on these big problems. Each country has its own, and each has subgroups within countries. Each one has their own agenda. But this is a global problem. It's not a Canadian problem. It's not an American problem. It's not a European problem. It's a global problem. And my hope is that I'm not one of these Pollyanna guys. I mean, look, I ain't going to be around when all hell breaks loose, but I would not buy land in Florida or I don't have enough money to buy land anyway, but if I did, I wouldn't buy it there or New Orleans or someplace like that. I mean, we're there, and it's a mess. And there's also the tornadoes, because you got more heat in the Gulf of Mexico, which is driving these things. You got tornadoes coming up all the way to Montreal, and you've never had that before. And we're all in this together. But my hope is, one of the things that it seems to me that we, particularly in democracies, do not do well, and that is to think ahead. And I will not speak for Canada, but I will speak about the United States. We do these things of planning 4 years ahead because that's Mr. Obama would like credit for this, or Mr. Bush would like credit for this, or whatever. They're a bunch of politicians. But you see, these problems are not 4-year problems. They're not even 40-year problems. They're 400-year problems. These are long-range things. And I believe that we do a very poor job in looking into the future and seeing the sorts of things that we know doggone well are coming. We are adults. We're educated. We're intelligent. We can see the problems coming. We got to come together. And I think it's the governments that the politicians they make the decisions. And I would like to see them put together some long-range, I'm talking 100 years, some long-range plans. See these problems. And it's not just this global warming. In Australia, if they had done this, where they had the chance, they would have waterways going up into central Australia, and it's still a darn desert. They didn't want to see the money. Now, they had a chance 50 years ago. They didn't do it. Now they're paying a price. And we're all going to pay the price if we don't attack this problem effectively and plan into the future. And the rest of it is just going to be remedial stuff. We're going to try to do our best to get through it. And understand, we Homo sapiens will, and about 300 years from now, temperatures will start to drop down, and we will have gone to green energy, and all that kind of stuff will have happened. But we saw it coming, we didn't do it, and that's shameful. Okay. I hope that helps. Yeah. I think I'm preaching to the choir here. Yes, sir. I'm sorry. I'll let them moderate. I don't know the rules around here.
I've got the talking stick. Well, first of all, Bill, thank you for a very entertaining and interesting talk. And I had a couple of questions about the sulfuridum microbes.
Sure.
First of all, is it possible, or I'm just thinking about the lack of evolution and plate tectonics and mutations and stuff like that, and I was wondering if you could speak to factors other than like environmental change caused by other biological factors or oxygen, or the Earth is moving, right? Like it's a constantly moving system, and I'm a little skeptical as to how isolated any environment can be. So that was my first question. And then the other one, which is probably the more interesting for the overarching theme of the colloquium, is if these things have anything in common with the other strings and beads and jelly beans. Like if there's any, if you think that we're looking at common
Sure.
ancestors or whatever.
Sure, both very good questions. Let me ball you out. Not you. Let me ball you all out, because this is something that I see, and throughout my life have seen repeatedly. And the reason I've seen it repeatedly is because it's prevalent, but it's prevalent and it's wrong. Geologists think in geologic time. Biologists think in biological, or what I call ecological time. And the two are vastly, vastly, vastly different. Geological changes, like movement of continents, are exceedingly slow relative to even species lifetime, but certainly relative to reproduction in organisms. But it's not just movement of continents. It is changes in the Earth's environment, the buildup of oxygen. Happens over hundreds of millions of years. But you see, the average species lifetime of an organism, of a given species, is about 10 million years. And the average reproduction of an organism is yearly. 98% of organisms on this planet reproduce once a year. They're tied to the yearly cycle. That means in 10 million years you had 10 million generations. 10 million generations permits organisms to adapt to these changes that happen over 100 or 200 or 300 million years. So we do ourselves a disservice if we intermingle those two things. And I've seen this repeatedly, say with regard to the origin of metazoans. Most paleontologists are in geology departments, and they say, 542 million years ago there was the first shelly organisms and so forth, and we think maybe 10 million years they diversified and so forth. But see, 10 million years is longer than it's 3 times longer than Homo sapiens has existed. 10 million years is an eternal time, and the changes in Earth's environment over geological time are so slow that organisms easily can adapt to them. In fact, I would go so far to say I think it is almost impossible to extinctify life on a planet once it gets diverse and adapted. Unless you had a way to vaporize all the world's oceans, which people have postulated, but I don't particularly think is true. But aside from that, the other, with regard to the cyanobacteria, you see exactly the same thing that I talked about here with the sulfuridum organisms in the Croacasians and the oscillatoriaceans and the entophysalidaceans and the nostocaceans, and that is well established, that I first discovered in 1968 and then put a name on it in 1994 after it had been found by 78 other scientists in 35 countries over 25 years. I wanted to make sure that I was right about it before I said anything about it, and put a name on it and codified it. Okay, so I was right, and everybody, and that's the way we name these things. I mean, you got a modern genus, Oscillatoria, and then we find something in the fossil record, we call it Oscillatoriites, which means like Oscillatoria, or Oscillatorioides, which means like Oscillatoria, or Eooscillatoria, which means dawn oscillatoria, or Archaeooscillatoria, and because it's oscillatoria, it just hadn't changed over time. How in the world can that be? Well, the reason is, and I would love to, but I don't have time to go into the genetics of the whole system, but what happens is that these changes in geology and the environment are so slow in the photic zone where these stromatolites are living. The generation after generation after generation after generation, remember day length is changing over this time too, and they all adapt to that. They adapt to, life is adapting very slowly, because it's biology. That's what they got to, you adapt or you die. So they didn't die, so they adapt, but they still have a genetic memory of how to take care of the problem before. So, for example, there was a very high UV flux early in Earth's history because we didn't have an ozone layer because there wasn't any oxygen. Guess what? Cyanobacteria are among the most resistant to ultraviolet light of any microbes known to man. Why? Because they were in the photic zone. They were going to get zapped by UV, so they got a bunch of UV repair mechanisms. They don't need those now, but it's left over from that early stage. There are lots of examples like that. So that's the way the system works. It turns out that when we think of evolution, I wrote a paper about this, disparate rates, differing fates, in Proceedings of the National Academy. The fact is that we think of sexually reproducing organisms like ourselves, like metazoans. These guys are all asexual. Their only source of genetic variability comes from mutations. And they're adapting all the time, but they got the genetic memory. Cyanobacteria can even do nitrogen fixation, which is an obligately anaerobic process. They can do that. They used to do it at night when there was no oxygen around. Then oxygen builds up. They can still, the modern ones can still do that if you put them in an environment where there's no oxygen. Because that's left over when there was a nitrogen crisis and they needed nitrogen to make amino acids to make proteins. I hope that's hopeful. There was some more questions. Yes, ma'am. Oh, hello there. This is an old friend of mine from UCLA of years and years ago, but I won't say how many years ago because she'd get mad at me. Go ahead. She's now a professor.
An incredible talk, as always, Bill. This is maybe a naive question, or maybe I missed something, but you were arguing that these forms from 2.3 billion years ago were very similar to ones from 1.8 billion years ago, and using that as evidence that there was no evolution during this time. But you also showed us some similar forms that exist now. And so if you extrapolate that argument, that would imply that there's been no evolution now.
I'm arguing that there has been no evolution in this group of, in those sulfuridum that are in subsurface muds at the bottom of the ocean at least since 2.3 billion years, up to the present. There's been no evolution in them at all. It's the same fabric, same organism, same environment, same thing.
Okay, so you were saying that during the first 2 billion years there was no evolution, but you're also saying that for the last 4 billion years in these organisms there's been no evolution. Is that right?
No, the only evidence I can cite is I can go back to the first occurrence that they're known. Remember, there's a difference between time of origin and first occurrence. First occurrence in the fossil record is at 2.3 billion years. I have one example at 2.3 billion, I have another example at 1.8 billion, and I have the present. My prediction is that as soon as these papers get published, people will look in these deep water facies, which nobody's ever looked at before, and there'll be half a dozen more, and it'll be exactly the same story, except I do think there are two organisms that I don't have that I think I'll find out when they came in later, because I do have them in the Miocene, which is just another story. But I don't know when the sulfuridums first came in. I do know that there were sulfate-reducing bacteria at least as early as 3.2 billion based on sulfur isotopes in pyrite. But I don't have any of these fossils yet earlier than 2.3. But remember, none of this stuff's been published, so nobody's looked for it yet. And as soon as we do, when it comes out, people get all excited and they'll say, oh, I can see that. Oh, there's one other thing I should say. that the story is going to get more complicated because I have stromatolitic assemblages from Brazil, from Australia, from a bunch of places. There are at least 7 or 8 that I know, in which I know now I also have sulfur bacteria at the bottoms of them. They're in the anoxic nuds beneath the photosynthetic zone above. And heretofore, when I found them, I named them bacteria incertacetus. That is, bacteria of uncertain biological affinities. But I haven't talked about it here, but there are a bunch of morphological characteristics that only they have, that cyanobacteria do not have. I've been puzzled now what they were, and now I know the answer. But see, that's another paper. I can only do so much.
Can I just continue a little bit then?
I think maybe just one more question. There's so many people, we're not going to be able to take all of the questions. So one more person on this side with a question, and then lots of opportunity at the reception after.
Hi. I was wondering if you had some sort of theory of how the sulfuridum got there, or got to be in those waters in the first place, if there was such an unchanging environment.
Question is, as I understand it, is how did the sulfuridum get there? Well, that's not too surprising. Let us remember that these are microscopic organisms, just like the cyanobacteria are microscopic organisms. Now they're spread by winds, by hurricanes, by wave action. Our the United States' U-2 spy planes will stick up sort of a catcher's mitt up in the stratosphere, and they've got cyanobacteria from these things as well. Not a whole lot, but enough. It's not too surprising these microorganisms have cosmopolitan distributions. So it's not difficult for them to get into an environment. And the other thing to think about in that regard is that, ask yourself, do you know of any sterile environment on this planet? One that is absolutely sterile, aside from, one of these operating places in a medical place where it's supposed to be sterile, which they're really not, but anyway. Look, life adapts. Life will, if it gets a toehold on any place, it will start, it'll grow. Even, look, in the land of the blind, the one-eyed man is king. You got an organism that is really slowly growing, but if there's enough nutrients for it to live, it'll live and do just fine, and then it'll turn out that, well, okay, there's some mutations that come along and it gets a little better at what it's doing. And that's the way evolution occurs. That's the way they adapt to things. So it's not surprising that they are there. It's surprising that in some ways they didn't change, but then that makes sense if the environment didn't change.
I also know they got there if the environment didn't change. I'm just confused about how they got there in the first place if it's such an unchanging environment. Because usually when you think of things being transported along, you're talking about winds and waters turning. That's what I'm
Well, okay. Today, for example, the organisms of this type. I showed you one example off of Chile, but if, to my knowledge, there are 23 stations going down to the tip of South America all the way up to Panama, in which this exact same sulfuridum occurs. And the fact is that, there might be a mudslide. there might be a mudslide, and then there is a current up above the water, up above the interface. They're down in the muds, but now they get shoved up, and now they get wafted up someplace else, and then they live there. And then there'll be a mudslide or turbidity current, and so forth. But where they are living is a stable environment that hasn't changed. Okay?
Nancy, over to you.
Well, I just wanted to express our great thanks. We have a thank you for you. In the tradition of the First Nations people whose territory we're on today, we got you a carved talking
great.
stick. It is carved with an image of the raven. If you want to take it out, you can see. And the raven holds a very important place in the mythology that sort of ties in with our emergence of life theme. And just for the benefit of those of you who can't read what's written on the box, I will tell you a bit about the talking stick. So for centuries, talking sticks have been used by the West Coast First Nations during potlatches and other ceremonial celebrations to symbolize the sacred power and honor to speak uninterrupted. It empowers the speaker to speak honestly and truthfully from the heart. The talking stick also serves as a reminder to the witnesses of their vital role of listening thoughtfully. When it is passed, the honor is then transferred to the next speaker. The talking stick teaches us to honor the sacred point of view of every living creature. So thank you again for your presentation.
Thank you.